Authenticating the true source of a raw cut of meat is a straightforward process, but when manufacturers boil them down, extract the fats, and blend them into complex commercial matrices like cosmetics or gelatin, tracing the origin of those ingredients becomes a severe forensic challenge. This technical diagram illustrates the baseline for targeted testing. Laboratories typically search for species-specific mitochondrial DNA using polymerase chain reaction, or PCR, to identify and amplify the distinct sequence of nucleotides that prove the presence of an animal source. That target is fragile. The intense heat applied during commercial processing physically denatures the double helix structure, tearing the genetic evidence into fragmented strands. Even if fragments survive the heat, complex product matrices introduce a second barrier. Compounds like polyphenols and polysaccharides act as chemical inhibitors, physically binding to the DNA and blocking the PCR primers from attaching. When the primers cannot bind, the test returns a negative result. The adulterant is still present in the product. The testing method simply failed to see it. If the single targeted biomarker is completely destroyed or obscured, analysts have to stop looking for isolated strands of DNA. They have to map the variants of the entire chemical environment. The standard forensic toolkit relies on finding a specific target. When that target degrades, laboratories must shift to an entirely different method of chemical observation. That alternative is chemical profiling. Instead of confirming one specific compound, profiling extracts the complete chemical fingerprint of a given sample. For an oil-based matrix, analysts extract the lipids using solvents and subject them to esterification, producing a complex mixture of fatty acid methyl esters. These esters are injected directly into a gas chromatography mass spectrometer for separation and analysis. This chromatogram plots retention time against mass spectra. Every peak represents a different fatty acid. A single run generates hundreds of data points detailing the concentration of each constituent. Cross-referencing these variations across many samples creates data too dense for human interpretation. To manage this extreme density, raw profiling data is processed through Multivariate Data Analysis, or MDA. Halal authentication no longer relies on finding a single needle in a haystack. The laboratory now mathematically analyzes the exact composition of the entire haystack. Raw datasets extracted from the spectrometer are notoriously noisy. Before statistical modeling can begin, the data requires rigorous pre-processing. Analysts first measure the sampling adequacy. A dataset must achieve a Kaiser-Meyer-Olkin value greater than 0.5 to prove the variable distributions are suitable for analysis. This data matrix illustrates how missing variables are standardized. Biomarkers that fall below the limit of detection are converted to a strict value, typically 0.5 times the detection limit, preventing blank cells from skewing the model. The algorithm then searches for outliers by measuring the Mahalanobis distance. This calculates how far an individual data point deviates from the mean center of its sample group. Any biomarker whose distance to degrees of freedom ratio exceeds 2.5 is flagged as a potential outlier and stripped from the dataset. Without this mathematical cleaning process, subsequent models will interpret lab cross-contamination or baseline instrument noise as actual chemical characteristics. With a clean dataset established, the variables are fed into a principal component analysis. PCA is a dimensionality reduction tool. It evaluates hundreds of individual chemical variables and collapses them into a few major axes based on their shared variance. This biplot displays the initial state of the raw dataset. Mapped along two principal components, the sample points lack clear boundaries. The analyst refines the model by isolating the significant biomarkers, specifically those with a factor loading greater than 0.75, which carry the most weight in differentiating the samples. By filtering the dataset down to only those high-impact variables, the revised biplot reveals immediate spatial organization. The chaotic scatter resolves into distinct territories, cleanly separating the sample sources into groups S1, S2, and S3. PCA successfully translates invisible chemical concentrations into physical geography, effectively cordoning off pure samples from adulterated ones. PCA is an exploratory technique. It provides a highly useful visual map, but it does not mathematically prove that the clusters are distinct. To validate the groupings, the data undergoes cluster analysis. which computes the precise numerical dissimilarity between the samples. This dendrogram visualizes that computed dissimilarity. Samples with highly similar chemical fingerprints are linked together at the bottom, forming larger branches as the variance increases vertically. By setting a statistical threshold, represented by the dotted horizontal line, the algorithm cuts across the vertical branches. The three resulting intercepts perfectly match the S1, S2, and S3 groups identified in the PCA. The visual clusters are now mathematically validated. The differences in the chemical profiles are proven to be an objective reality, not a statistical illusion. With the distinct chemical boundaries proven, the laboratory has a functioning model. They can now test an entirely unknown commercial sample. Analysts use discriminant analysis, a predictive technique designed to categorize new data against the validated groups. This classification matrix demonstrates the performance of the DA model. It achieves 100% correct classification rate in placing the samples. Fisher distance tests show p-values falling well below 0.01, giving the laboratory absolute statistical confidence that the sample groups are completely distinct. The laboratory then introduces the unknown dataset into the model. The algorithm instantly maps the new profile, locking the sample into its respective cluster. Once the sample is classified as adulterated, regression analysis steps in to calculate the exact percentage of contamination present in the matrix. Analysts run multiple models and select the one with the lowest root mean square error, ensuring the lowest probability of an incorrect calculation. These predictive models establish quantitative certainty. Even in processed materials where DNA fragments have been entirely degraded, the laboratory can still achieve a conclusive authentication. These computational methods ultimately serve the bench-level analyst who has to issue a legally defensible testing report. Testing facilities must adhere to ISO 17025 standards, which dictate that this entire profiling and modeling pipeline must be formally verified before it is applied to commercial products. Many analytical methods fail this standard because they are researched in clean isolation. True validation requires proving the method works accurately inside the chaotic matrix of real-world retail samples. Relying on a single strand of DNA is a vulnerability in modern manufacturing. Authentic assessment requires looking at the total composition. By utilizing multivariate data analysis, forensic laboratories extract signal from the noise, providing undeniable proof of origin regardless of how heavily a product is processed.